Welcome, dear students, to today's class on antidiuretic hormones. In short, it's called ADH. So, we've been taking continuous sessions on pituitary hormones, and this is the last one of the pituitary hormones. ADH, or the antidiuretic hormone, is also called vasopressin, or more conveniently, it's called as arginine vasopressin. It's a peptide hormone of 9 amino acids, and the amino acid list is as given: cysteine, tyrosine, phenylalanine, glutamine, asparagine, cysteine, proline, arginine, glycine. And there's a disulfide bond between the two cysteine residues. So the hormone is synthesized as a prohormone in the hypothalamus, which travels to the pituitary and is released from the pituitary. The function of the vasopressin is to reabsorb water in the kidney. It can constrict arterioles, thereby increasing the arterial blood pressure. And it's also important in social behavior, sexual motivation, and bonding. The half life is 16 to 24 minutes. So, as I said earlier, the ADH and oxytocin is synthesized as pre-pro hormones. So, each prohormone contains the structure of the oxytocin or the ADH and a co-secreted peptide, which is either neurophysin 1 or neurophysin 2. If it is ADH, you can see the presence of associated neurophysin 1, and if it is oxytocin, you can see the presence of neurophysin 2. So you can see it over here. You have the exon 1, intron 1, exon 2, intron 2, exon 3. So in exon 1, there is a signal peptide which would be cleaved. So the N signal peptide is cleaved on transport into the ER. And then you have the hormone and the neurophysin. Right? And this is a glycopeptide. So pre-pro vasophysin is the name for the pre-pro hormone of vasopressin, and pre-pro oxyphysin is the name for the pre-pro hormone of oxytocin. So the hormone or the peptide that has been synthesized is packaged in the endoplasmic reticulum and the Golgi apparatus, and finally into membrane-bound secretory granules. And the secretory granules, through an ATP-dependent transport mechanism, transports the hormones inside the secretory granules to where it has to be released. So during this transport, the prohormone is proteolytically cleaved to the hormone, and the neurophysin part is removed. So, removing the signal peptide converts the pre-prohormone to prohormone, which happens in the ER. And after that, when the prohormone is packaged into secretory granules, an ATP dependent transport mechanism comes into place to transport the prohormone in the secretory granules to which region of exit or a release from the cell. So during this transport, the prohormone is proteolytically cleaved to form the hormone and release the neurophysin. So what is the function of vasopressin? So we'll consider the yellow part of the pathway where the water content of the blood is normal. So consider you drink, I'll just take a marker. So consider you drink too much of water. What happens is your water content in the blood becomes high, and the brain produces less ADH because you have enough of water. So when the brain produces less ADH, less volume of water is reabsorbed by the kidney, and the urine output the urine output becomes high, right? And the water content of the blood comes back to normal. So consider a state where you have had too much salt in your food, or you're profusely sweating in a humid climate, and suddenly your water content in your blood goes very low. So that signals the brain to produce more ADH, and as a result, a higher volume of water is reabsorbed by the kidney, and the urine output becomes low, or it becomes concentrated because the water content in the urine has been reabsorbed, right? And your water content comes back to normal. So this is the normal process of the function of ADH. So ADH acts through two receptors, the V1 receptor and the V2 receptor. The V1 receptor is important for increased systemic vascular resistance, and the V2 receptor is important for increased blood volume. And both of these processes cause an increased arterial pressure or blood pressure. So the binding of the ADH to its receptor, as we saw right now, V1 or V2 receptor, activates adenyl cyclase. You can see it over here. So it activates the adenyl cyclase, which converts ATP to cyclic AMP, which activates protein kinase A. The protein kinase A activates what is called as AQP. So AQP is also known as aquaporin. So aquaporin is a water channel or a protein involved in the formation of water channel, which helps in maintaining the concentration of water or the reabsorption of water. So the pKa phosphorylates AQP2, which is an aquaporin protein, by and increases transcription of its gene by the cyclic AMP response element. So the vesicles that contain phosphorylated AQP2 then moves towards the apical membrane. This is the apical membrane. Moves towards the apical membrane, and it is the movement is towards the apical membrane is driven with the help of molecular motor called dynein. So in this figure, you can see that ADH is binding to V2 receptor. It's a G protein coupled receptor. After the adenyl cyclase, ATP is converted to cyclic AMP. Cyclic AMP activates protein kinase A. Protein kinase A phosphorylates dynein, phosphorylates AQP2 in secretory granules, which helps in the process of transport of the AQP2 containing secretory granules towards the apical membrane. So dynein is a molecular motor which actually drives the movement of AQP2. And when it reaches the apical membrane, the secretory granule fuses with the membrane and releases the AQP2. Snares, which interacts with vesicles and facilitates the fusion of these vesicles with the membrane. So snares are proteins which allow the release of secretory granules or contents of the secretory granules. So the addition of ATP to the membrane allows the water to enter the cell driven by the osmotic gradient. In the absence of ADH, the ATP2 water channels are removed from the apical membrane, by what is called the clathrin-mediated endocytosis. So the AKP2, what you have to understand is that the AKP2 is a protein which can form a water channel on the apical membrane, which helps in reabsorption of water based on the osmotic gradient. When the AKP2 is absent, the water channels are absent and water cannot be reabsorbed. So AQP2 water channels are removed with the help of clathrin-mediated endocytosis. And the endocytosed AQP2 is either stored in cytoplasmic vesicles for reinsertion or into the or it's degraded, right? This is the process where this is the process of exocytosis. It fuses with the cell membrane, and after the water reabsorption has been completed, it can again be endocytosed into the cell, which can be recycled or it can go for degradation. So, regulation, increased osmolality of extracellular fluid. Example is increased salt intake, that is ADH, increases ADH release to dilute ECF again to normal. Decrease in blood volume, example, hemorrhage conditions. There is a decreased frequency of inhibitory impulses from volume receptors in heart to the supraoptic nuclei, so that there is increased release of ADH. Renal ischemia due to hemorrhage causes increased renin concentration, which leads to increase in angiotensin 2, which ultimately leads to increased ADH secretion and increased water reabsorption. Alcohol inhibits ADH secretion, which causes diuresis or an increased urinary volume output. So, natriuretic peptide or ANP inhibits the ADH secretion by inhibiting the angiotensin 2 induced stimulation. So, this process, renal ischemia due to hemorrhage, this process, like there is an increase in angiotensin 2 leading to an increase in ADH secretion. This process is inhibited in the presence of atrial natriuretic peptide. Sixth one is the anxiety, pain, and trauma. Administration of morphine and narcotic, and even hot weather can cause an increase in ADH and a corresponding increase in water reabsorption. So this was about ADH hormone.